Hello everyone. In this lecture we're going to talk about single gene inheritance. So up until this point we have talked about all of the cellular mechanisms that we need to understand as far as how traits are passed from one generation to the next. So we started at a very micro level, looking sort of the smallest aspects or components of genetics that we can. We're going to move up now. So we're going to move up a level and we're going to talk now about traits themselves. And remember, a trait is, once again, referring to a gene. In this case, a gene is a segment of DNA. And we already understand how the process of protein synthesis works in order to make the proteins then that exhibit those traits. So we're going to talk a little bit more about how patterns of inheritance were discovered using the work of Gregor Mendel. We'll talk about single gene inheritance patterns, the law of segregation, independent assortment, which again align with some things that we have already discussed. So first of all, let's start backwards. Gregor Mendel is typically considered the father of genetics. So he is the one who is given credit for discovering that each individual possesses traits. Those traits, copies of those traits get passed on from one generation to the next. And what's interesting about Gregor Mendel is he was actually a monk. And this is not uncommon that you have people that are, in the clergy or represent their religion that turn out some really interesting scientific discoveries, simply because this is something typically that they're free to have the time to do in their other pursuits related to their religion. So Gregor Mendel, in addition to being a monk, was a really brilliant mathematician, and he was an avid gardener. He loved plants. He had some beautiful gardens, and he was really, really interested in understanding things like probability and chance, and so that those two interests kind of merged together, and he was able to use his interest in botany and mathematics, and he was able to determine how traits were inherited. What's interesting is he was well ahead of his time. So he wrote all of this up, published all of his work, and nobody knew really what to make of it. They didn't understand it, more or less. So for about 50 years, no one really noticed his work at all. And it was sort of well past his death that someone finally was doing the work that he was doing, that started to do research and figured out that Mendel was the one who actually had gotten there first. So he worked with this particular plant, which we're going to talk about. These are pea plants, this your regular average garden variety pea plants, and they have some really great characteristics that make them really important when it comes to studying genetics. And the reason why pea plants were chosen is that they exhibit two distinct forms for a variety of different traits. So seed form, color, pod form and color, the position of the flowers, the seed coat color, the length of the stem, the flower colors themselves. So there's a whole host of traits here that he was able to follow. And it was important that there were only two distinct traits, right? Because this way you could follow that trait very clearly. If you had 3 or 4 distinct forms, it would get entirely too complicated. So the point here was following these two distinct forms of these traits. All right. So we consider Mendelian genetics, when you hear this term, Mendelian genetics, it really is a pattern for inheritance that is followed for single gene traits with distinct alleles. Okay. Now remember, alleles, excuse me, I apologize. My throat is all groggly this morning. Remember that alleles are forms of a gene, right? So an allele is a form of a gene. So a pea plant that has yellow pods versus green pods, for example, the gene is for pod color and the alleles are green or yellow, right? Those are just alternate forms of a gene. That's what an allele is. In Mendelian genetics, it's one gene, it's got distinct alleles, and we can follow those patterns. But to be honest with you, most inheritance is not this straightforward. Most time you have multiple genes working together, you have impacts and influences of other things like the environment on the traits. So there's only so many genes that follow this sort of Mendelian pattern of inheritance. All right, so let's do a little bit of vocabulary review before we get into the meat of this. So again, a genotype is your combination of alleles. So what forms of those genes do you have? If you have, we typically denote these with letters, which I'll talk about in just a minute. If you have two of the same type of allele, you're going to be homozygous for that trait. If you have two different alleles, you are going to be heterozygous for that trait. So going back to the peas for a minute, and let's go to the pea pods. If you have two green alleles, that means you're going to have green pea pods, okay? And you are homozygous for that trait. If you have one green allele, one green version, and one yellow version, you are heterozygous for that trait. And the color is going to depend on whether the trait is dominant or recessive, which we'll talk about in just a minute. So your genotype is the combination of your alleles. Your phenotype is the physical outcome of that genotype. So again, green pods, green seeds, short plant, purple flowers, whatever it happens to be. And your phenotype depends on the nature of your alleles. It depends on whether you have dominant or recessive alleles. So traits are either dominant or recessive. So let's talk about that. A dominant trait is any trait that can mask the presence of one allele can mask the presence of another allele, okay? A recessive allele is one that is masked by a dominant allele. So an easy way to think of it this way is for a dominant trait, you only need one copy of that allele in order to physically see the trait. For any type of recessive trait, you have to have two copies of that allele in order to see the trait. So the easiest way to imagine this is by using letters. So for alleles, we use letters to denote alleles, okay? So in this case, we have, let's talk about tall pea plants for a second. So for tall pea plants, we are going to use the capital letter T. T stands for tall. Being tall is a dominant trait. For the same trait, the opposite allele is short, but rather than using a new letter like S, that would just get confusing, we just use the lowercase letter for the same letter that we're using. Okay, so capital for dominant, lowercase for recessive. T for tall, capital T, little t for short. All right, so that is just sort of the background that we need to continue on and understand what Mendel did in his experiments. So here's what he did. The first thing Mendel needed to do is he needed to produce what are called purebred or true breeding when it comes to offspring. The idea that when you take two individuals, the offspring will always look like you for that trait, no matter how many times you do that cross. So going back to tall plants, for example, if you took two tall plants and got all tall offspring, and you did it again and again and again, you would know that those plants have been true bred for the trait of being tall. I can also give you the example of white flowers here. If we take two plants that have white flowers and they only will produce offspring that have white flowers always, we know that those plants have been true bred for white flowers. This is important because what Mendel was trying to do is he was trying to create homozygous individuals. So again, homozygous individuals are those individuals that have two alleles that are identical. So taking short parents and always getting short offspring, taking tall parents and always getting tall offspring. This is how we can make sure that the plants are homozygous for the traits that we desire. So once he had that done, he had all of these different plant stocks and he had a whole host of different true breeding parents. The next thing was to start doing some crossbreeding. So crossbreeding is we're going to take a trait like flower color, but we're going to take the two different forms of that trait. So we're going to take the true bred form for, let's say, flower color, purple, and we're going to cross that with the true bred form of flower color for the other color, in this case white, and we're going to see what we get. So in this example, we take purple flowers and we take white flowers, we cross them together and we end up getting all purple flowers. So what does this tell us? it shows us which traits are dominant and which traits are recessive. So back to our tall plants and short plants here. When we cross a true-bred tall plant and a true-bred short plant, we get all tall plants. So what that means is that we have heterozygous individuals. They have one tall allele and one short allele, and it tells us that in this case, the dominant trait is being tall. So our tall allele is dominant and our short allele is recessive. So this is how Mendel was able to figure out which traits were dominant and which traits were recessive. So this led him into his first his big principle of genetics, and this is called the law of segregation. And the law of segregation says that the distribution of alleles of a gene into separate gametes during meiosis. Okay, so this is not new to us because we have already talked through the process of meiosis, so we know exactly how meiosis works. But when Mendel did his work, no one had any idea what he was talking about. They didn't have alleles. He called them units. He called them separable, distinct units for traits. And you got one from each parent. And the idea was that gametes only contain one allele for each trait, and only one observed, version is observed in the individual. The other allele is there. It's still present, but we just don't actually see its expression. So again, this should look very familiar to you because this is what happened when we were talking about meiosis, right? So we already know this, but again, this is before we knew anything about cellular division. This is before we knew anything about DNA at all. So Mendel was way ahead of his time in this discovery. Okay, so let's talk a little bit about some handy tools that we can use to predict genetic crosses. This is called a Punnett square. And a Punnett square is a predictive tool. We can use it to predict probabilities that offsprings will have particular genotypes and phenotypes. So as long as we know what the alleles of the parents are, we can use that to predict what the offspring are going to look like. So remember that a parent has two alleles, okay, and those alleles are going to then separate into gametes, right? So a homozygous individual can only produce a gamete with a single type of allele. A heterozygous parent can produce two possible alleles, right? So what we do is we put the gametes that the parents can create on the top and the left side of a 4 square, and then we fill in the squares with the genotypes of the offspring, and that will tell us what the phenotypes of the offspring will be. So let's take a really simple example. We're going to use those true breeding parents for a moment. So we've got those true-bred tall parents. They will have 2 alleles, but the alleles are identical. They're all capital T's. So this is parent 1 at the top. Parent 2 is on the left-hand side. All you do is you combine the T on the left and the T at the top for each box, and that tells you what the offspring will be. So when we do all of these combinations, top and left, all of our offspring have a 100% chance of being genotype big T, and a 100% chance of all being tall. That's a pretty easy one to follow. Let's do one where we're actually looking at a cross. So in this case, we're going to cross a tall parent with a short parent. Things look a little bit different now because with a tall parent and a short parent, we still have all of our offspring looking the same and having the same genotype, but now their genotype is 1 tall allele and 1 short allele, right? So your F1 generation, F1 just stands for filial. Filial is a way to say offspring without saying offspring. So P1 is parent 1 generation. F1 is filial, just means your first generation of offspring from those parents. 100% chance of being tall, and 100% chance of having a heterozygous genotype, big T, little t. Well, let's make things even more interesting. What happens when we cross 2 parents who are heterozygous for a trait and not homozygous for a trait? This is called a monohybrid cross. So hybrid meaning you have 2 different alleles, okay, so you're a heterozygote. Mono meaning we're only doing this for a single trait. So now things get a little bit more interesting. Parent 1 is here at the top, parent 2 here is on the left. Now we have different chances for different genotypes, right? We only have 1/4 of the offspring are going to be homozygous dominant. 1/4 of the offspring are going to be homozygous recessive, and half of the offspring is going to be heterozygous as far as genotypes go. So here we go, right? 25, and how do I do 1/4? 1 out of 4. Okay, so this is a probability square. So there are 1, 2, 3, 4 squares. 1 square out of 4 is 25%. Okay, 2 squares out of 4 is 50%. So that's where my numbers are coming from. And then when we look at phenotypes, oops, sorry, let me go back up. I didn't mean to do that. Well, we can do it here, I guess, as well. Sorry. No, I can't do it here because now I'm looking at a dihybrid cross and I didn't want to go to a dihybrid cross. Here we go. Back we go. What I wanted to point out is that the genotype or the phenotype is 75% tall and 25% short, right? Because remember, these heterozygotes will exhibit the dominant trait. So I just wanted to make that note before we moved on. Okay, we can also do this for 2 genes. Okay, so we can also take, in this case, let's look at seeds. We have round yellow seeds, which being yellow and being round are the dominant traits, and we can cross that with a wrinkled green seed, which are the recessive traits. If we're doing crosses that are true bred, okay, it's easy to follow this because we can just follow the dominant trait, and we know that all of the offspring will be heterozygous for both traits. So that's fairly straightforward. And we can again do this with a Punnett square. And again, it works when you have simple combinations, like again, so this is a dihybrid cross for 2 homozygous traits. Again, when you're homozygous for a trait and you only have 2 different combinations that you can combine here, so RY, capital little RY, it's fairly straightforward. And again, all of the offspring are going to exhibit the dominant traits in this case. What this shows you, though, is what's important to note is now we're going to move on to what's called the law of independent assortment. So the law of independent assortment says that 2 genes on different chromosomes will segregate their alleles independently. So what does that mean? It means when you inherit one trait or one allele for a particular trait, it does not influence which other allele for the other trait you're going to inherit. So the idea here is that traits don't follow each other. Okay, so you have an equal chance, again, depending on if your parents had those alleles to pass on, you have an equal chance of being tall and having purple flowers as you would of being tall and having white flowers, okay? So traits don't follow each other. Now, there is a little bit of a caveat to this because there are something called linked traits, which we'll talk about a little bit later in the semester. But as far as Mendel was concerned, traits didn't follow each other. They weren't inherited together. They were inherited separately. So again, this is also a review from meiosis. We know this. The law of independent assortment came from Mendel, okay? This is how we got genetic diversity, right? So crossing over and independent and random alignment, independent assortment, here it is in action, and this is why it's important. So with that information, what happened if we had a dihybrid cross and we had heterozygous traits? So what if you had 4 different gametes that you could produce for 2 different parents? Yeah, we could do a Punnett square for that, but that would be a little bit ridiculous. So what we do instead is we use probability, because remember, these are based on these squares, 4 squares, right? And so we can use probability and product rule to predict the genotypes for offsprings of more than one trait. So if I wanted to find out the probability of obtaining a plant that had wrinkled green peas, that's the recessive, from 2 dihybrid parents, so dihybrid means they are heterozygous for both traits, how could I figure this out? Well, you figure each trait separately. So there's a 1 out of 4 chance to be homozygous recessive for R, and if you need to review that, go back to the slide where I showed you that. And then there is also a 1 out of 4 chance of being homozygous recessive for the Y trait. So you just multiply those two together, okay? 1/4 times 1/4 equals 1/16. Now, does this hold true? Yeah, let me show you how it holds true. Let me go back to the previous slide. Oh, that'll take too long. In any case, if you go back to my previous slide that had the great big Punnett square on it, you would see that this holds true because there's only one square that contains, out of those 16, that contains an offspring who has this particular genotype. So this is a lot faster and a lot easier. All right, so from this lecture, you should remember these terms: homozygous, heterozygous, dominant allele, recessive allele, true breeding, and crossbreeding. And you absolutely should know how to use Punnett squares, product rules, and know both of Mendel's laws.